Good evening, everyone. Welcome you all to week 12 NPTEL TA session, which is on ethical concerns, germline gene editing. So, please let's wait for 5 minutes and let other people join. Then we'll start our class. Just wait for 5 minutes. We'll start from 6:10. Okay, let's start. Please let me know if my screen is not visible to you or if you are having any problem with my voice. Okay. So today we'll be talking about and we'll be discussing some questions on ethical concerns, bioethics. So first thing, what is bioethics? Bioethics is the study of ethical, social, and legal issues that arise in biomedicine and biomedical research. So here we are in this uh, NPTEL class. Here we have been studying about genome editing, and we also talked about different types of genome editing tools like CRISPR-Cas, ZFN, and talents also, and it can be implied for different types of organisms, right? So here comes the ethical point of view in which organism can it can be used and how much the research purpose can be used. Okay, and it includes focus on issues in healthcare. Just a minute. Issues on healthcare like how if any patient has having any problem with their single nucleotide polymorphism or like that, and any because of any point mutation, like for example sickle cell anemia. If a sickle cell anemic patient wants to be cured by CRISPR-Cas technology or any type of gene editing method, then also if the methodology provided by the researcher or the scientist, is it ethical or not, that also includes bioethics. Addresses ethical issues in public health. This the example comes in case of public health also. In case of public health, how we are treating different patients and how the researchers is dealing with the samples. Like generally, uh, for clinical research, researchers or scientists collect various types of blood samples or different types of tissue samples in a mass or in a number of people, and then research on them and then found the critical result they want from them. So there are also ethical rules how much and how can be this is used. Okay, that also includes bioethics, and it also focuses on issues pertaining to relationship between human activities and environment, how human activities is connected to environment, and how this biomedical research is playing its crucial role in all these things. That also includes our ethical rules and concerns. Focus issues in conduct of research, right? So research should be conducted in a manner so that it won't affect or it won't hurt our overall microenvironment or bioenvironment inside which we are living in. And bioethics generally includes genetically modified organisms, biotechnology or nanotechnology, organ trafficking. Okay, specifically in our last session, we have been talking about this organ on chip, right? Using a chip method, we can develop, we can just test our different medications or drugs on different types of liver tissue or brain tissue or cardiac tissue or any other. For that, we don't need any particular organ to test a particular sample. We can go for organ and chip method so that reducing the number of killing of mice and other type of animal models. Practicing integrity of participants in therapeutic trials, that is I was talking about. Environmental ethics. Environment means environment includes everything including plants and animals. They are animals, they can talk, and plants also. We just can't just put our research output in on them and look out for a result. There is also bioethics rules so that we need to follow it as a researcher for understanding and getting our output result. Includes biodiversity, human cloning. We will be talking about human cloning in detail, associated procreation, and others. Okay, and also access to medicines and healthcare services, healthcare devices. So generally happens that people in India also, we can get medications from a normal drugstore chemist, from the normal chemist at our neighboring street, right? But in case of different countries like US and others, they don't provide you medication without giving without you providing any prescription, right? That's also includes bioethics. Here in India, we need to implement that thing, and we need to go for prescribed medicines only. That would be greater be helpful for reducing the different types of medication related side effects. So this all over everything concludes our bioethics. Let's talk about it more. What's on the horizon of bioethics? What we need to go for study? Why we are studying this? Everything is written here. You can go for it in detail, but I will be talking about it shortly. That is beginning of life, health and society, data and technology, animals, food and environment, research ethics. So beginning of life, that means there are specific research that require embryo, okay, human embryo. They want to try it on human embryo so that they can get specific results, and that drug can be used for treating different types of carcinomas or different types of mutation related disorders. And for that, in case of different types of labs also, they have shown good results for testing their samples on embryos. But not in many cases we can't use them without having any ethics or not. Health and society, we have talked about it. If you are giving anesthesia or any kind of uh, medication to that particular patient, the doctors need the uh, this paper to be signed by their authorized person, so that they can go for the transplantation ho ya fir any anesthesia or whatever it is. So in case of health and society also, bioethics is there. Data and technology, how much data can be used, including biohacking also, medical implants, biohacking. Animal food improvement, for example, is GMO, right? Genetically modified organisms or genetic modified fruits and vegetables has been in market, but we can't just create one genetic modified fruit or everything and just open into to the street market. It needs to be go for different types of trials and all, that if it has side effects or not, so like that. It includes animals, food and environment also. Research ethics like brain surrogate, fetal tissue research, and everything needs our research ethics. This all over, I just briefly talked about everything in shortly, so you can go for it via looking at the slides. So this is about bioethics, what we are talking about, and we'll be doing more questions. At first we will go for the history, who started bioethics and how it comes to a picture, and after that we'll go for different types of human genome editing and different types of editing technologies where bioethics can be used. Okay, so this is our first question. The term bioethics was coined by Fritz, a Protestant pastor, philosopher and educator in Haller and Dele, in which year? So at first we'll go for the history part little bit, then we'll move to our modern bioethical problems. Please try to answer this question in 2 minutes. Write in the chat box. If you don't know, you can guess also, then we'll be discussing about it in detail. Okay, let's talk about it. The term bioethics was coined by Fraser in 1926. So your answer is 1926. Fraser published an article entitled Bioethics, a review of the ethical relationship of humans to animals and plants, and proposed a bioethical imperative in 1927, extending Kant's moral imperative to all forms of life. Right? So, what he did, he just published one article named Bioethics, a review of ethical relationships of human to animals and plants. So, what does it mean? That humans are just evolutionary, superficial, and improved to as comparison to animals and plants. But it doesn't that mean that we can implement anything without hurting those animals and plants. Okay, so we need to go for different ethical principles to do research on them. He evaluated contemporary physiological knowledge and the ethical issues surrounding the emergence of pluralistic secular society. In the process, Fries it reinterpreted moral obligation toward both human and non-human life forms by defining bioethics as an academic field, a guiding principle, and a virtue. Okay, so here he tried to give moral support to those animals and plants which have been used for research without any purpose. Like we can't just use their number of different types of animals for our research and kill them one by one. This is against social morality. So here he tried to connect both human and non-human life forms by as an academic field by a moral support. So this is all about bioethics term. Let's move to another question. Who introduced bioethics as global ethics? Here are the four options. Please try to answer this question. Okay, let's talk about it. When Rensselaer Porter, an American scientist, introduced the idea of bioethics as global ethics in his 1971 book, that is Bioethics: A Bridge to Future. With his extensive and lengthy experience in cancer research, Porter presented a novel interdisciplinary idea with the aim of fusing ethics and science. He intended to create a conversation between the science of life, biology, that is bios and life, and usable knowledge, philosophy, ethics, values as a result, and he coined them the term bioethics. So he tried to connect biology with philosophy and termed it as bioethics. Potter's bioethics united humility, responsibility, and multidisciplinary and intellectual competency. So here is your answer. Okay, let's move to another question. The Asilomar conference was a milestone for science because it was the result of days proposed by the scientists themselves. Bioethics, biosafety, self-regulation, or ethics. Please try to answer this question. So let's talk about it. In 1975, 140 scientists, doctors, and legislators gathered in a Asilomar Conference Center in State Beach, California, in order to debate the ethical implication of genetic engineering. Okay, so This ethical implication of genetic engineering, it was started in 1975. Certain principles of biosafety were established at this conference with the object of preventing an accidental leakage of recombinant organisms which could affect human beings, animals, or environment. The Asilomar Conference was a milestone for science because it was the result of self regulation proposed by the scientists themselves. At this conference, the scientists agreed that research with recombinant DNA should proceed, but appropriate safeguards should be outlined. So, what is the reason behind this? Why they used self regulation? And it was proposed by the scientists themselves only. So the reason is general, that is, if you leak accidental leakage of this recombinant organism into the society, like may for example into the animal world, or into the environment, or it may affect the human beings also. So sometimes what happens, this recombination leads to harmful mutations, right? And these harmful mutations can sustain a generation after generation if this mating proceeds and offsprings being generated. Okay, so and we don't want that. So that for that reason also, and also for another reason, if we talk about different plants and agricultural point of view, if we are introducing any recombinant plant or any vegetable to the market, without any trial methods, then it may affect the animal or human being which is intaking it, or it may change their genetical in genetical in a genetical mode, that is in the change in the chromosomes may lead to different types of mutations and harmful effects. That's why there are different certain reasons why the scientists proposed the self regulation, and it was a great success for science. Okay, so everything for this ethical implication of genetic engineering was started in 1975. Let's move to our next question. So in the case of heritable genome editing, there are two distinct concerns. Select the right option. The possibility of off-target effects of the editing process, as in the case of somatic gene editing. The intended genome edits themselves might have unintended sequences even in the absence of off-target effects. Some worry that, taken to its extreme, germline editing could create classes of individuals defined by the quality of their engineered genome. Please read this question twice or thrice anytime you want, and try to answer this question in 2 minutes, then we will be discussing it. Okay, then let's discuss about it. So here in the right side picture you can observe how genome editing can be used for and can be done, like using meganucleases, zinc finger nucleases, FOK1 is the restriction enzyme used, TALEN, CRISPR-Cas9. We have discussed about everything. So all these cutting machines like zinc finger nucleus, TALEN or CRISPR-Cas9 genome editing tools, they cut the double-stranded DNA at a particular site, making it a double-stranded break. After that, it followed by non-homologous end repair, like including insertion or deletion or group disruption, and it also can be followed by homology directed repair, providing a precise DNA editing gene insertion. Okay, so here using homology directed repair we can do the insertion thing, and using NHEJ, that is non-homologous end joining, you can perform the insertion or deletion related studies. So our question was about heritable genome editing. So the possibility of off-target effects of the editing process, as in the case of somatic genome editing. Whatever standards are developed for somatic applications, and there will be less tolerance for off-target effects in germline application. Off-target effects, do you understand? Off-target means there are two types of target, that is on target and off target. So if we are trying to make a cutting at a particular DNA site, but it's not cutting at that site, and the cutting is been happening at other site rather than the target site, then it will be called as off target effect. The intended genome edits themselves might have unintended consequences even in the absence of off target effects. In the case of heritable genome editing, to convert a well understood disease causing variant gene to a widely occurring non-pathological variant, the editing change would be to a version of the gene that is known not to have deleterious consequences. So this answer will be 1 and 2. Okay. So here is another question. Which one is not a principle for the governance of human genome editing? Promoting well-being, providing the whole protocol to the individual, transparency, responsible science. There are certain principles are present for governing the human genome editing project. So please try to answer this question. Okay, let's discuss it. So, the principles of human genome editing includes promoting well-being. That is, the principle of promoting well-being supports providing benefit and preventing harm to those affected, often referred to in the bioethics literature as the principle of beneficence and non-maleficence. Transparency includes the principle of transparency requires openness and sharing of information in ways that are accessible and understandable to stakeholders. While due care, the principle of due care for patients enrolled in research studies or receiving clinical care requires proceeding carefully and deliberately and only when supported by sufficient and robust evidence. The principle of responsible science underpins the adherence to the highest standards of research from bench to bedside in accordance with international and professional norms. So, here is your answer, that is providing protocol. Which amendment deals with the production and management of human embryos? Dickey Wicker Amendment, Fritz Walker Amendment, Gilbert Amendment, or none? Please try to answer this question. Okay, let's talk about it. Dickey-Wicker Amendment states that none of the funds made available in this act may be used for the recreation of human embryo or embryos for research purpose. Research in which a human embryo or embryos are destroyed, discarded, or knowingly subjected to risk of injury or death greater than that allowed for research on features. For purposes of this section, the term human embryo or embryos includes any organism not protected as a human subject under 45 CFR C46 as the date of the enhancement of this act, and that is derived by fertilization, parthenogenesis, cloning, or any other means from one or more human gametes or human diploid cells. So, which amendment deals with production of and management of human embryos? That is Dickey-Wicker Amendment. The main technical problems with safety implications for potential clinical ZZE in human embryos revealed therein include the following option except one, which is fairness, mosaicism, off-target effects, or on-target undesired modification introduced within or next to the targeted locus. Everyone, please try to answer this question in 2 minutes, then we will be discussing it. Okay, let's discuss about it. Safety issues associated with germline genome editing. The main technical problems with safety implications for potential clinical germline genome editing, that is ZZE, in human embryos revealed therein includes Mosaicism, a situation where not all cells of an embryo or organism have the same DNA. In this case, the desired DNA modification can be off-target effects where unintended changes in the genome outside of the targeted sequence can occur, while undesired modification introduced within or next to the targeted locus. So here in the right side figure, we can observe how this germline genome editing can be done. So just a minute. Here it is showing a male individual and using the testis biopsy report and affected spermatogonia acid derivation followed by CRISPR-Cas9 editing in spermatogonia acid gamete differentiation. Now the sperms have been corrected. Now it is showing a healthy sperm. For example, using sperms also affected sperm leading to in vitro fertilization or ICSI, followed by CRISPR-Cas9 genome editing, and that leads to correction of the embryo. In case of male or female, here it is showing any skin biopsy, that is, it can be liver, it can be kidney, or it can be any other organ, providing reprogramming of iPS derivation, that is induced pluripotent stem cells, and CRISPR-Cas9 genome editing in case of IPS, that is gamete differentiation followed by to corrected embryo. In case of females, it can be used as conventional stimulation affected oocyte or GV oocytes followed by IVF and CRISPR-Cas9 gene editing, ultimately leading to CRISPR-Cas9 editing embryo or corrected embryo. Okay, so this is the method of germline gene editing. Germline means this oocytes or these are called germlines. That means the egg or the sperm, they are called germs. Okay, so this germline genome editing provides us a corrected embryo. So if we know that any mutation is happening because of this specific gene sequence and it can be corrected in the germline only, then it is followed by different types of protocols, and that leads to correction of the embryo and after a healthy individual. Okay, so let's move to next question. As per a December 2018 poll, Americans draw the line at so-called enhancement but favor the use of genetic engineering to address disease and disability, clinical research ethics, cancer, or oocyte procurement. Everyone please try to answer this question. Okay, let's discuss about it. As per December 2018 poll, Americans draw the line at so-called enhancement but favor the use of genetic engineering to address disease and disability. Okay, so here is their answer is disability. Which diseases and disabilities to target, however, is still an open discussion. Some questions that may help inform this decision that includes: should there be a focus on infectious disease resistance? Only fatal conditions should be included or not? Will we decide that there is a need to quantify the degree of suffering? If an effective treatment already exists, should we still seek prevention through genetic modification or not? If the childhood versus adulthood onset of illness an important factor. So these are the different important factors to address for genetic engineering. And in 2018 December, the Americans draw a poll regarding these questions. Okay, so let's move to our next question. He Jiangqi shocked the world when he broke the news that his team at Southern University of Science and Technology in Shenzhen, China, has successfully edited embryos using CRISPR-Cas9 and delivered two baby girls. On which date? January 25, 2018, November 15, 2018, or November 25 or December 25? Please try to answer this question. Okay, let's discuss about it. It was on November 25, 2018. Okay, so this is the method of CRISPR-Cas9 mediated genome editing, where it is a wild type oocyte. The zygote, in case it is in S phase, and CRISPR-Cas9 genome editing can be used here and leads to wild type embryo. But if we introduce one mutation here using this CRISPR-Cas9 tool, then that also leads to different types of untargeted mutant embryo, mosaic embryo, like wild type mutant or wild type non-homologous end repair mediated insertion or deletion mediated or homologous directed repair. Okay, so this can be different depending upon the uh, uh, genome we are targeting or which CRISPR-Cas method we are using. Okay, so this is the last question of today's session. The DES is one of the about 30 genetically modified organisms to sidestep the USDA regulatory system in the past 5 years. Banana, BT cotton, BT brinjal, or mushroom. Everyone, please try to answer this question. Okay, so let's talk about genetically modified organisms. So mushroom is one of the most 30 genetically modified organism to sidestep the USDA regulatory system in past 5 years. In each case, the agency's Animal and Plant Health Inspection Services has said that the organisms, mostly plants, do not qualify as something the agency must regulate. Once a crop passes the USDA reviews, it may still undergo a voluntary review by the US Food and Drug Administration. Several of the plants that bypassed the USDA were made using gene editing techniques such as the zinc finger nuclease and transcription activator like effector, that is talent systems. But until now, it was not clear whether the USDA would give the same pass to organisms engineered with science's hottest new tool, that is CRISPR-Cas9. Okay, so here in the level figure you can see that we are talking about genetically modified organisms and what food we can consider. There are about federal regulators have approved 11 GMO crops for sale in United States as of 2019. That includes corn, soybeans, cotton, alfalfa, sugar beets, canola, papaya, summer squash, innate potatoes, pink pineapple, non-browning Arctic apples. Okay, so these are the GMO foods has been approved by United States of until 2019. So next another question arises, are GMOs safe to eat? So the FDA regulates the safety of GMOs and all commercially available GMOs in the US have been through a consultation process and received FDA approval. And GMOs are safety tested. It is unlawful for anyone to market unsafe the food. GMOs are tested to avoid the addition of known allergens. Okay, so why we need to use GMOs? To increase the productivity, like ease of production, reducing the cost of production, and improves crop quality. To increase the food access, that is increasing yield, that is equal to increased availability, make food more appealing, reduce food waste like that, and reduce carbon footprint, reduce fuel uses like using cotton, reduce the use of toxic pesticides, improve groundwater quality. And nutrition and medical enhancement include develop insulin, address nutrition deficiencies. So, uh, so this is all about today's assignment, and we have discussed about 10 questions on bioethics. I think it will be helpful for everyone to do their assignments for this week also. I hope, and this is the last TA session of genome editing method. So I hope everyone will do better in their exams, and I wish them all the best. So if you anyone have any question, then you can ask me. If you have any questions, you can ask me. Okay, if you don't have questions, then we can end this session here. And I wish you good luck for your upcoming exam. Thank you for joining.